Hello, everyone. Welcome to today's webinar, Virus Specimen Preparation in Biosafety Level 3-4 Laboratories. My name is Stephen Goodman, and I'm Chief Scientific Officer of Microscopy Innovations. I'm excited to be hosting this webinar and pleased to be able to present today's keynote speaker, Candace Blancett. I'll provide a more detailed intro in a few minutes. Before we begin with the technical content, I'll provide some general info about the webinar and about Microscopy Innovations. First, today's webinar will be available on demand after this live session via the Microscopy Innovations webpage under Resources, where you can also access related content such as the citations we referenced today. Secondly, we'd love to hear from you with any questions you have during today's presentation. You may send them to us using the question and answer button on the left side of the screen and enter the question and click Ask on the bottom of the screen. We'll be answering questions at the end of the session. If we don't get to your question during today's webinar, we'll follow up with you afterwards. And of course, you can always ask us questions via the website links. Thank you. And last, we'd like to encourage you to share today's webinar with your colleagues via social media once it's posted online. Once again, I'm Stephen Goodman, Chief Scientific Officer for Microscopy Innovations. The agenda is shown here. I'll first provide a brief introduction about our company's products and then provide a very brief story about how this whole relationship began with Candace Blancett, our invited speaker. And then I'll introduce her. Following her presentation on these two topics, we will answer your questions, which you can ask at any time, again, using the question and answer function. So let me provide a quick overview of the Microscopy Innovations and the mPrep system. Our mission is to create better tools for microscopy specimen preparation, to eliminate or reduce tedious and repetitive tasks, improve reproducibility, improve preparation speed, enable automation. Overall, our goal is to make workflows efficient for the microscopy laboratory. Let's begin by taking a look at the mPrep system specimen workflow, which is shown here for a typical transmission electron microscopy protocol. So at the very beginning of the protocol, a specimen is removed from an experimental animal or wherever your specimen comes from and placed into an mPrep S specimen capsule. It is then entrapped, and at this point, the specimen is never touched again, so it's only touched at the time of insertion. The capsule is attached to an ordinary pipetter, a pipetteman, and the pipetteman delivers all of the chemical reagents to the specimen from possibly the initial fixation, certainly through dehydration, post-fixation, and resin embedding. Once the specimen is fully embedded in a resin, it is transferred to the oven for curing. After the specimen is now fully embedded and cured, the entire capsule can be clamped into the microtome chuck for sectioning in pretty much the usual manner. Once a specimen is sectioned, the sections are placed on grids in the usual manner, and these grids are then inserted into mPrep G grid capsules. The grid capsules, like the specimen capsules, are then attached to a pipetter for all the reagent exchanges in the same manner. Now, what I've shown here is that all of the sample preparation is one pipetter and one changing reagents for one capsule at a time. The reality is most laboratories will use multi-channel pipetters, either the 8-channel shown here or a 12-channel here, to do multiple specimen preparations at the same time with the same or with different reagents. However, for the greatest efficiency and speed, all reagent exchanges, rinses, and agitation can be done under automated computer control using the mPrep ASP1000 automated specimen processor. This enables essentially any protocol while you do other things, and this provides the ultimate in laboratory social distancing, something that obviously is important these days. Let me provide a couple of examples of this before we move on. So I'm going to show examples for specimen processing and grid processing. So this first example comes out of the University of Maryland, Baltimore, and it's using the ASP1000, the automated processor, for immunogold labeling of tissue specimens. So this is a pre-embedding or en bloc immunogold labeling protocol. This second example is of a fairly standard tissue sample preparation, in this case of kidney, and this was done automated. We see TEM, and we see a light microscope image of a kidney biopsy in this case, and this was done at the University of Wisconsin. This particular sample used very rapid reagent exchanges for the processing, which one can do with the ASP1000. And enabled this entire preparation to be done, all the reagent exchanges for this preparation to be done in about 2 hours. So speeding up the preparation quite a bit. These next two examples are serial block face, three-dimensional electron microscope imaging. The first one is our flatworm cells from the Stowers Institute in conjunction with HHMI, and the second one was at the Cleveland Clinic Foundation of brain, looking at synapses and synaptic structure. This latter preparation was done roughly in 24 hours, again, using the automated sample preparation, which represented a considerable time saving compared to a conventional manual preparation, which would take the better part of a working week. I'm now going to show just two examples for grid processing. The first one is automated processing. Again, it's immunogold labeling, in this case of bacterial flagella whole mounts, again, done at the University of Maryland, Baltimore. This particular laboratory, the University of Maryland, Baltimore, often does immunogold labeling on grids overnight, which then helps to increase their workflow and productivity. And this second example shows manual processing of negative stain sample of bacteriophage T6 out of the University of Toulouse in France. This provides a good prelude to today's virus story. So how did this virus preparation story begin? Well, today, I'm speaking to you while we are in the midst of the COVID-19, the SARS-CoV pandemic, which is arguably the worst pandemic that's occurred since the 1918 H1N1 influenza. Unfortunately, zoonotic epidemics like SARS-CoV-2 are not all that unusual. In the last 2 decades, we've seen epidemics from dengue, another SARS, and influenza H5N1, Zika, Ebola, to name just a few potential, unfortunately, relatively smaller epidemics that did not become pandemics. This story began with the 2014 outbreak of Ebola in Zaire, I believe. So at this time, some of you, I'm sure most of you will remember this one, it was a pretty scary time. We see the cover of Time magazine. We see a patient arriving at Emory University in Atlanta. We have the World Health Organization tracking this. And while this was going on, my phone rang. And on the other end of the line was Dr. May Sun from USAMRIID, the U.S. Army Research Institute of Infectious Diseases. And that's me in the other picture there. May explained to me that their technicians working in biocontainment destroy or damage 80 to 90% of grids. She wondered if we could help. And of course, I answered, of course. We can eliminate all grid handling in biocontainment. So together, I suggested that we develop a protocol for negative staining that will work in biocontainment. And with that, I'm going to move and introduce our speaker who will describe how biocontainment sample preparation, negative staining of viruses is done at USAMRIID. So our invited speaker is Candace Blancett. While I'm introducing her, I'm going to let you know that I will be passing the electronic baton to her to take over the presentation. So it's possible that you will see some blank screen for a little while. The audio will remain, and then Candace will take up the gauntlet and present. So Candace is a lab technician at the United States Army Medical Research Institute of Infectious Diseases, usually called USAMRIID or USAMRIID, in Fort Detrick, Maryland. Quite a place. She has 8 years experience working in BSL-3 and BSL-4. Diagnostics is definitely within her capabilities. She's a trainer in biocontainment procedures, electron microscopy, and histotechnologist. She's the author of 6 publications, and just a couple of months ago, at the end of this last semester, she received her master's in biomedical science from Hood College, just a couple of months ago. So, Candace, please take over the baton.
Sorry, that went a little faster than I meant for it to. So as Steve said, thank you guys for coming, and thank you, Stephen, for having me here to speak. I appreciate everybody's time today. I've been at USAMRIID for about 8 years, and some of that time has included working in the electron microscopy lab. So I'm going to share with you some work we did, including the mPrep capsules into our negative staining procedures, and then also a virus quantification protocol that we updated. So the first thing I'll present is on our negative staining. This is a picture of the building USAMRIID. We are one of the few institutes across the United States with BSL-4 capabilities. If you're not familiar with the different biosafety levels, this first picture here is biosafety level 2, which is probably what you're familiar with. Technicians are usually just wearing a gown over top of their regular clothing and then one pair of gloves. This lady in the middle here is working in BSL-3, so she's wearing, at USAMRIID we wear scrubs. Not all institutes with BSL-3 do that. Some people do wear their normal clothing underneath of a Tyvek suit in BSL-3. Anytime you're working with live agent, it has to be in a biosafety cabinet, and you're going to have to be wearing 2 pairs of latex gloves. And she's also wearing a respiratory unit. It's called a PAPR. And just so you guys are aware, for coronavirus, USAMRIID is handling all of our live coronavirus in a BSL-3 environment right now. And then this fellow all the way to the right is in a BSL-4 suite, so he's got on a positive pressure suit. He's got on a pair of gloves that are attached to his suit, which are actually quite similar to the gloves you wear when you're doing the dishes at home. So if you can imagine trying to wear those kind of gloves when you're doing lab work, it can be very difficult, and it greatly reduces your tactile sensibility. And then he also has on another pair of gloves that he can change in between samples and keep his hands clean. So kind of what Stephen said, the reason we started looking into the use of the mPrep capsules is because a task such as pipetting in the BSL-3 and BSL-4 suites is very easy, but a task like grid handling can actually be quite difficult. You're fairly far away from your sample. You can't get as close and to see things as close up as you can in BSL-2. You might have some restricted vision. It's a little bit hard to see directly in front of you because the way the suit or the PAPR shields are. You tend to have at least 2 pairs of gloves on, so that can be a little bit restrictive in your movement as well as how well you can feel different things. And then lastly, we usually try to minimize the use of sharps as much as we can in BSL-3 and BSL-4. And since the tweezers that we use to handle grids are quite sharp on the end, that was a tool that we were interested in trying to minimize the use of inside biocontainment. And then, as I mentioned previously, any live agent we work with has to be handled in a biosafety cabinet. I'm hoping that you've worked in one of those before, but there's continuous airflow, and it forces you to be a little bit farther away from your sample than you might be on a regular laboratory benchtop. So an issue we have commonly with biosafety cabinets and working with EM grids in them is that you can easily destroy your grid if you just manipulate it wrong. It's easy to accidentally fold it up or crush it somehow. It's a little bit hard to control how quickly your reagents dry out if you're trying to do something like negative staining out in the air. And then last but not least is a lot of times we just lose them. If you've worked with EM grids before, they're very lightweight and small, and they can kind of get sucked up into the air vents and things like that. And obviously once that happens, your sample or your grid is just gone at that point. And then all the live agents that we work with in BSL-3 or BSL-4 have to be completely deactivated before they're allowed out of the suites. So we have 2 different approved protocols that we use at USAMRIID in order to pull out electron microscopy samples. There's 24 hours of exposure to 2% glutaraldehyde, and we usually do this with samples still in their liquid form, and we just mix 1-to-1 the sample with 4% glutaraldehyde. This takes longer because you have to allow for 24 hours of fixation before you're allowed to remove it from the suite. And it also requires more than one entry by a biotechnician, a biocontainment technician. This may not sound super major, but I can tell you when I go into BSL-4, like literally just to get in and then back out of the suite, it takes me about an hour, and that doesn't include time doing anything. So it can be quite time consuming to go into the suite, consecutive entries to get something done. It also requires the dilution of the sample in the fixative, which may not be a major issue, but sometimes we're dealing with samples where the original person who created the virus stock may not be really sure about the concentration of the virus to begin with. So we're a little bit averse to diluting things up front because sometimes it's easy to dilute your sample beyond where you can really beyond the limit of detection for electron microscopy. So what we usually try to do is get our sample onto a grid, and then we use 1 hour of exposure to 1% osmium tetroxide fumes to deactivate our sample and fix it. This can be samples can be removed from biocontainment on the same day, so you just have to have an hour worth of exposure to osmium tetroxide, and it doesn't require you to dilute your sample up front if you choose not to. And just a little bit of a recap on the mPrep grids, the mPrep grid system that Stephen talked about. You can fit the grids inside of a pipette tip is what it looks like. There is a filter you can put on here. The filter's not shown in this picture. You attach it to the pipette, and it's as simple as pipetting from there. This is just demonstrating staining and some washes. Here's some up-close pictures of the mPrep capsules. You can fit 2 grids into the mPrep capsule. Loading the grids into the capsule is the hardest part of using them. As an EM tech, I was able to figure out how to do this pretty quickly with just a little bit of practice. This is definitely something that we do in the EM lab, is load these grids into the capsules, and we may give them to another technician outside of the EM lab who has access to more samples than we do so that they can use them, but I wouldn't really ask somebody not experienced with grid handling to try to load the grids into these capsules. The slats are labeled. If you can see over here, there's an A and a B, so if you do need to differentiate between the 2 grids within the capsule, you can do that. It takes about 35 to 40 microliters of reagent in order to cover both grids. It's a sealed system for staining, so that does help with safety, but you should be working in a fume hood or anything you need if you are working with hazardous materials. There's a cap which can close over top of the capsule. It doesn't touch the grids when it's closed, but it can give you a fully contained little safe place for your grids to live. You can actually use this system for long-term storage if you choose to. As you see down here, this person has labels on their system. We did not do that at USAMRIID because we had another system already in place for grid storage prior to this, but it's not a bad idea if you don't already have a previous system set up. And this is just to walk you through the protocol that we used at USAMRIID. So in the BSL-2 electron microscopy lab, we would load 2 grids into each mPrep capsule. The filter is shown here. This can be attached to the mPrep capsule, similar to like if you're using a filtered pipette tip. So we usually gave the technicians that needed to operate the pipette the filters to go with the mPrep kits. And they're going to take that into the biocontainment lab. So this is an example of a man working in BSL-4. So for the negative staining protocol, all he had to do was take out his virus stock. He's going to aspirate 35 to 40 microliters of stock into the pipette tip. He's going to allow the sample to adsorb to the grid for about 10 minutes. You can do this either vertically or horizontally. Most of the examples you're going to see here, we were doing it where we would lay the pipette tip down horizontally. And that was really just because you have to have some sort of rack or something in order to keep the pipette tip vertical in the BSC, and we didn't always have availability for that. So it was just easy to lay it down, and that's what we ended up doing. After the 10 minutes, we would rinse 3 times in DI water and then fix in 2% glutaraldehyde for 20 minutes. So this is just going to fix the sample a little bit and make sure it help keep it adhered to the grid. And then after the 20 minutes of glutaraldehyde, you would eject that, and we would put the capsules containing the grid with the sample on them in a 50 ml conical tube. So at the bottom of this tube, we would have a piece of filter paper that was soaked in osmium tetroxide. So we wanted to expose the grids to the fumes, but we didn't want a lot of osmium tetroxide liquid floating around this tube because that can actually stain your sample further upon contact. So we came up with this idea to soak filter paper and put it in the bottom of the tube so that we could get the fumes in the tube, but not have a lot of liquid floating around. And after an hour, we would take this back to the BSL-2 EM lab. So we are now cold side, just in normal clothes and just wearing one pair of gloves and a lab jacket. We would remove the mPrep capsules, attach them. We would use a new set of filters. We usually left the original filters in waste in BSL-3 or BSL-4. We would rinse the sample again with 3 times with DI water, and then we would follow our negative staining protocol. So we usually used either 1% uranyl acetate or 1% phosphotunsic acid, and different technicians had a little bit different luck with different times. I preferred UA for 30 seconds. There were a couple different technicians I worked with, and we all had slightly different protocols. If you work in the EM world, you probably have done a little bit of negative staining and have your favorite reagents down to the exact seconds you like them. We would take a piece of filter paper and blot the grids still in the capsule dry, and then we would let it air dry a little bit beyond that, and then go on over to our transmission electron microscope for imaging. So just to review the protocol in BSL-2, we were using 200 to 400 formvar grids with carbon coat on them. We would load 2 of those into an mPrep grid, and the filter would also be provided to the technician going into biocontainment. And we also provided those technicians with all the reagents. So we would give them a conical tube with 1% osmium tetroxide soaked filter paper, DI water if they didn't have access to it in the suite, and then the 2% glutaraldehyde they were going to need for that 20-minute fix. And in BSL-3, the user simply has to aspirate the virus, rinse 3 times, and bring it out to us in that conical tube with osmium tetroxide. And then on the BSL-2 in the electron microscopy lab, we were going to rinse again and stain for 10 to 60 seconds with either UA or PTA, and then blot dry with filter paper. It says or air dry. In my experience, you really want to make sure you remove the stain with filter paper, because if you leave it on the grid for too long, it can allow for the stain to continue to seep in and you end up with positive stained samples. This is also why we did not have a lot of luck. We did experiment with allowing the biocontainment technicians to try to negative stain on the hot side and then bring the sample out to us. And we really didn't get very usable samples that way. And I think a lot of the issue was it was very hard for them in all their gear and in the BSC, and everything's a little bit farther away and harder to see, for them to use the filter paper to like sufficiently pull all of the stain off of the grid. So I think it was sitting on there longer, and we were getting too many positively stained samples that you really couldn't get the contrast you needed for good imaging. So in our experience, it's a lot better to let them do put the sample on the grid hot side, but bring it out and do the staining at BSL-2 by the EM technicians. So using this, we had about 90% success rate with like not having mangled or lost grids. This was a huge improvement for us over our previous experiment. We had different groups who tried to do EM preps with us. And it was very difficult because we'd get back these formvar grids that were just completely destroyed. They'd be balled up in a little ball, and you'd pull them out and be like, oh God, there's probably no formvar left on this thing and no sample. And it was just really difficult, and it was very frustrating for the people who were trying to do the preps to give us the virus so we could look at things for them, because they'd be spending all this time hot side trying to do these preps and have nothing to give us either, and it was really frustrating on both ends. We found the results were comparable to the droplet method, and the biggest leap and bound for USAMRIID was it did enable preps by people who aren't really trained with grid handling. So I guess something I might want to explain about USAMRIID before we keep going is that we work with biological select agents and toxins. So those are different agents that have been deemed as a very high risk and to safety by the government. So the reason that it's kind of hard for us to have access to all the viruses that different groups we work with want us to image is because these are all tracked by the government. They have very restricted access on who's allowed to access different things. So even though I have a group who might come to me and say, hey, I want you to image this Ebola for me, I can't get in there and get out the Ebola virus stocks and set them up. So that's where we have to do these collaborations where I rely on maybe a virology technician who typically does virus stock, like propagates virus and maybe works with animals normally, and I need to give them EM grids and ask them to get a sample on a grid for me, and it's very difficult for them if it's not something they do every day. So within USAMRIID, it's just really made collaboration a lot easier. We get access to more specimens, and more people are able to rope the EM lab into their projects. It gives you kind of the potential to try some different staining techniques and different deactivation compound, or we can use different deactivation methods, as I talked about. We can do the 2% glutaraldehyde or the 1% osmium tetroxide method. So again, we had a very good success rate with getting intact grids where we were able to see a lot of the different form bar squares were still intact, and there's lots of virus available for us to try to find that perfect virus particle to image. We did do a lot of comparing the mPrep method to kind of our standard droplet method. So to do this study, we did do the 24-hour glutaraldehyde fix and pulled the actual liquid virus stock out of biocontainment. And in our BSL2 lab, we did some droplet method staining and some mPrep method staining for comparison. This sample we were using is a strain of Ebola virus, and we were really quite happy with the quality of staining for both samples. We got the contrast we needed. You can see the little glycoproteins on the sides of the virus and the nucleocapsid. And we felt like the quality was quite equivalent between mPrep and the droplet method. This is chikungunya virus, and we just were comparing the 2% glutaraldehyde fix with the 1% osmium tetroxide. There really isn't a significant benefit to the quality of the images after either fixation. As I said, we tended to go back to the 1% osmium tetroxide fixation to remove it from the suite, just because we could work on the project faster. It was a little bit easier. It didn't require as much effort on the part of the technicians prepping the samples for us. The only thing you do have to be careful of is sometimes the osmium tetroxide can lend a little bit of positive stain to your sample and minimize the contrast a little bit. It's not something we've had to battle a lot, but it is something that has popped up now and then. And then this was just comparing negative staining with phosphotunsic acid versus uranyl acetate using the mPrep capsules. And again, it's no significant difference between the two. To me, negative staining is a little bit up to the technicians and the protocols you like. If you've been negative staining for a while, you might have a preference on which stain you like better and what percentage you prefer to work with. I personally always liked 1% uranyl acetate, but I did work with another lady who made really nice negative stains using phosphotunsic acid versus, for me, in my experience, I never felt that my results were quite as strong with phosphotunsic acid. So I do feel like that's a little bit of a personal preference and finesse thing. Some of electron microscopy is just that way. I think it's a little bit of a difficult science, but we try to do the best we can to get some reproducible results here. So in summary, we had about 90% successful preparations in BSL3 and BSL4, which was a huge improvement over what we were getting before. Preparation is a lot easier using the mPrep capsules. You're just pipetting versus trying to do the droplet method. We got equivalent results compared to the droplet method. It made it a lot easier for us to have inexperienced EM technicians doing the sample prep, which was typically required depending on the virus we were trying to work with. It adds an extra layer of safety where we're able to minimize the use of like one more sharp object, which is the tweezers that you use when handling grids. And we were able to get pretty consistent results using the mPrep system, arguably more consistent than when you're trying to prep in BSL3, BSL4. You can do consistent good preps in BSL2 using the droplet method, but it really wasn't that way for us when we were trying to work in biocontainment. And then it's kind of been able to be applied to different studies. We can use both types of stains if you'd like. We have a couple different deactivation protocols. And then we did take this into a different study, which I'm going to go into here in a minute, where we used the mPrep capsules for preparation of samples for virus quantification that was pretty highly requested among USAMRIID, and it made a huge, huge difference in that protocol as well. So I'm going to talk about STEMVQ, which just stands for Scanning Transmission Electron Microscopy. Virus quantification is what the VQ stands for. So we basically took an old virus quantification method done by EM and tried to make some updates to it to make it a lot more reliable. This was a method that was requested often by Dr. Cynthia Rossi down here. So she did a lot of Ebola work, and a lot of her stocks were used for animal studies. So they would try to quantify their stocks using plaque assays. They have a virus-site virus counter, which is a flow-based method to count. They basically stain the virus for genetic material and protein, and the virus site's going to count particles where it picks up both those signals. Um, they do quantitative PCR on their virus stocks, and they also like to try to do electron microscopy counts on their virus stocks because we could give them morphology data, and then we also have the ability with electron microscopy to count non-infectious or incomplete particles, which there are some published works out there that indicate the quality of your virus stock, even if it's not necessarily all infectious particles you're dosing your animals with. If there are uninfectious particles, that can affect the outcome of your animal study. So that's information that a lot of people across the Institute are interested in. But a few years before we revamped this protocol, she actually published a paper that was comparing all of these methods. And long story short, for the EM protocol, she basically came out and said it's very unreliable. It's not very helpful because it's so unreliable, and she wished it was better, but it's just really bad. So that kind of initiated all of our work in trying to revamp this protocol a little bit. So the first thing we included was the mPrep system, which we've talked about quite a bit, but it minimized grid handling. So the short and sweet of it is the mPrep capsules gave us much more usable grids, which gave us a lot of data that we didn't previously get out of the prep, the BSL-3 and BSL-4 stock preps. And that really made a huge difference for us. We also, in conjunction with the mPreps, we were working with a newer microscope for USAMRIID that allowed for a lot of automated imaging. So we could save a whole lot of technician time on imaging. We imaged more parts of the grid than we used to with the old protocol. And so the more data you have and the easier it is for everybody, basically just makes a much more friendly protocol to the technicians. And then we also started using automated software to try to count the beads in the virus when we could. So that also saved a lot of technician time and increased our data output. This is just another recap of the mPrep grids. We've been over a lot of this. If there is anybody out there who hasn't worked with EM grids, this is just a cool little picture I took for another talk where this little gold circle is an EM grid, and this white circle is actually the hole punch from a piece of paper. So if you think about trying to do anything with a hole punch paper in like a windy environment, that's kind of what our BSL-3 and BSL-4 technicians are fighting against. So that's why they were struggling so bad, and mPrep made quite a big difference for us. For the virus quantification, what we're actually doing is mixing gold beads with a known concentration. So we've had the concentration verified. It gets verified by the producer that creates the beads, and then we also would have it verified again by an outside lab just to confirm. And then the technician would mix the gold beads 1-to-1 with a virus stock, which we don't know the concentration of. And then that's what they're going to aspirate onto the grid. So the protocol in BSL-3 and BSL-4 is quite similar. They're just going to have the virus and the beads mixed together instead of just plain virus. You're going to aspirate that into the mPrep capsule and let it absorb for 10 minutes. Rinse 3 times with DI water. You're going to do the 2% glutaraldehyde fix for 20 minutes. And then we preferred, again, to use the osmium tetroxide vapor deactivation. You could, I suppose, do the 2% glutaraldehyde in your original virus stock and fix it for 24 hours and then pull it out, and we could do the prep in BSL-2 at that point. But again, you have a little bit narrow window of limited detection and whether or not we'd surpass that by diluting it. So we chose not to do that with most of the stocks. And then in the BSL-2 lab, we were imaging these on the scanning microscope because that's where we could do all the automated stuff with the microscopes we had access to. So we would rinse the sample 3 times in DI water. We dehydrated the sample on the grid through an ethanol series. We would send it through the critical point dryer. We would sputter coat it and put a 6 nanometer iridium coat over everything. and then we would image that. We had a Zeiss Sigma is the scanning transmission electron microscope we were using for this. And we were able to automatically set up the Zeiss so that it would scan images that were 35 microns by 35 microns, which is about the size of a form bar square on a 400 mesh form bar square. And we would tell it to take 30 images kind of scattered around the grid, so we would try to get an even representation of the entire grid. We limited it to 30 images per grid because we did a study at the beginning of this where we took one grid and we imaged 100 different squares on it, and we had our statisticians help us with all the math on this one, and they basically told us that by the time you hit about 30 images, you had about minimized the error across the grid that you were gonna. So you had about as accurate as you were going to get with 30 images, and continuing to image beyond that really didn't give you a whole lot of benefits. So that's where we cut off for our protocol. This is just an example of the image. We used ImageJ automatic software. Let me go back one, sorry. If you're looking at this, these smaller black beads that are very nice and round, these smaller black dots that are very nice and round are your 40 nanometer gold beads. And then these slightly larger circles you're seeing here are actually virus particles. So this is, in this example, I'm not sure which alpha virus it is, but it's either Eastern, Western, or Venezuelan encephalitis virus. They all look the same under the EM scope. We worked with a couple different strains on this study. So we were able to tell the ImageJ that we wanted to look for 40 nanometer circles that were about this density, and you can see the beads have now turned into white dots, and that's what it looks like when ImageJ is counting that as the object you're looking for. So we would run that program again and count for slightly bigger, slightly less dense to get the virus and plot those numbers against each other. And we would then use the slope to extrapolate out the original concentration of the virus because we know the original concentration of the bead stock. So we were able to calculate it out. And this is just an example of 3 different strains of alpha viruses we used, where we ran a dilution series and we counted them using our electron microscopy method. And we were able to get a linear range between like 10e to the 9th and 10e to the 12th, or sorry, 1e to the 9th and 1e to the 12th. So that's a pretty good indicator that we are getting accurate quantifications. If you're not seeing that linear change as you dilute your sample, then that's a pretty bad sign for your quality of quantification there. And this is just an example of conditions that lead to inaccuracy. So the picture all the way to the right is actually a nice sample prep. The beads are quite spread out, the virus is spread out evenly, and you're going to get a nice cluster of samples where they make a nice linear graph and everything. And your R number is going to be pretty clean on that. Versus the 2 to the right, you've got a whole lot of virus clumping going on in this middle one. You're probably hitting the upper end, or you have passed over the upper end of your linear range that I showed you before, and it gets really difficult to start counting virus like this because they get all clumped together. You can't really accurately say how many virus particles are in this little clump and this clump and this clump. So that's what makes your linear range really go off track once you get above a certain concentration of virus. And then this one is actually a lot of background and sediment. I think it's probably salt and stuff like that from the buffer the virus was in. So we just got a lot of background kind of dirt on this grid, and that interferes with both my ability and ImageJ's ability to kind of accurately count these things. So you're going to get a graph that looks more like this on those kind of sample preps. You can tell a lot about your accuracy just by looking at your graph. If you've got a lot of outliers, you probably don't have the best quantification data. This is 3 different virus stocks that we quantified. So what you're seeing with these 2 different columns is we actually quantified both grids in the mPrep capsule because we just wanted to verify that we were going to get the same quantity off of both grids that were used during the same prep, because if not, that's a pretty big red flag there. But we did actually get very similar results, about as good as science gets. And then We did find that the most significant contribution to error was variation between individual preparations. So these were the same virus stocks that were just prepped at different times. Some of them were a little bit more, there's a little bit more error versus this guy was a little bit more accurate. And we were within about a log for all of our samples, though, which was a huge improvement over previous methods we were using. And this is just comparing plaque assay data, virus site data, and the electron microscopy method for the same stocks. So we did find the electron microscopy numbers are these red squares. So for each stock, they were higher than the plaque assay and the virus site numbers, which we did expect. Because as I mentioned earlier, the electron microscopy method, we can count non-infectious particles or incomplete particles, and you're not going to pick that up in a plaque assay, and you may not pick it up in virusite as well. But what we did see was a similar trend where, you know, stocks that had a lower concentration by the other methods also had a lower EM method versus this one had the highest concentration. We also found the highest concentration. So that trend was very promising for us. And this is just kind of the acknowledgements to everybody who worked on this. So we had the pathology lab working on it. Dr. Cam Robinson had left before this study started, but he was an EM scientist that initiated a lot of the automated imaging that we worked with. The statisticians did a lot of the data analysis or helped us with a lot of data analysis. Dr. Rossi and her group were kind of the drivers behind updating the system, and then the viral biology group was amazing, and they prepped so many alpha virus preps for us because we were trying to get grids and data and grids and data. So they were very, very helpful in the work that we did with the virus quantification. And now I think we're going to move into questions.
Yes, definitely. We are ready for some questions. Candace, first of all, thank you so much for presenting. This was very informative, and hopefully our audience found it informative as well. So we have a couple of questions that have come in. Let me begin with a comment that several people made is, what do you hope to gain from this webinar? And I would say about half the people who signed up said that they were seeking information about or specific to processing in BSL-3 for biocontainment. And Candace, I think that was addressed extremely well. So first question for you, Candace. Actually, there are three that are basically pretty similar. If you could talk about how virus samples are deactivated differently in BSL-3 and 4 compared to BSL-2 lab. And one question was asking about how to deactivate SARS-CoV-2.
Okay. I'm going to go ahead and go back to this slide, which is our deactivation procedures. So we do have SOPs that we have to follow in USAMRIID for anything that we bring out of biocontainment. So we have two SOPs for electron microscopy samples, and we have to use this 2% glutaraldehyde for 24 hours exposure or 1% exposure to osmium tetroxide for kind of monolayer samples on a grid like that. We do have a different protocol for tissue, and it's been a while since I did tissue, so I'd have to double check, but I think that one is 2% in glutaraldehyde, but I think they have to stay in the sweep for a week if I'm not mistaken. I'd have to review that though if anybody wants that information. So these are like proven methods that we know the virus can no longer infect anything. It is dead, but we are trying our best to protect the morphology on the EM side. So these are the two protocols we use at USAMRIID. For coronavirus, we are treating it as a BSL-3 agent at USAMRIID, and we have to follow all of the biocontainment procedures with it in order to pull it out. So again, we would be using these same ones for electron microscopy samples for, like, I do some diagnostic type work too, where we do DNA and RNA extractions and things like that. So we are using lysine agents. One that we use a lot is Trizol, if you guys are familiar with that. That lyses the virus and it kind of leaves the nucleic acid available for use.